So today I will be showing you how to maintain the practical copy for class 10. This is the project for class 10 general science. So first of all you have to buy this type of practical copy. The cost of this type of copy is 60 rupees. Now on the first page you have to write your name here, then your class, your roll number and the name of your institution. So and you don't have to write here anything, and here you have to write the year and the subject name and the date when you will submit it. You have to write that date. Now on this page you have to write um, date. First of all you have to write the date on which day you are writing this, then the serial number and experiment number and the experiment name. Like here the first experiment is the study of natural indicators, and it starts from page 1, so you have to write the name of the page 1. And the second practical when you have done, if it is on the same day, you write on the same day. Like 25 December you have wrote both of the practicals, so you have to write here 25 and here also you have to write 25. Then the serial number will be 2, experiment number will be 2, and the subject or experiment will be here the experiment is anatomy and reproductive biology of hibiscus flower. You have to give this type of titles here in a concise way. And the page number, it starts from page 4, so you have to give here page 4. Okay, now let's move. Now you will get this type of pages here. You don't have to do anything here. Now don't start from this page because this page is not empty. You have to get this page empty, so don't use this page. You just move to the next page. Now when you will get this type of page where one side is plain and one side is ruled, from this page you have to start, and this page will be your page number 1. So first you have to give here page 1, then date on which day you are writing, and experiment number 1. Now first you have to write, first of all you have to do the experiment, then you have to draw your experiments. Like here figure color changes of natural indicators in different media. Now you have to do in 9 test tubes, but you don't have to draw all the 9 test tubes. You just have to draw the main test tubes. Like here we have drawn turmeric plus soap solution and colored it with reddish brown color. Here china rose and lemon juice you have colored here dark pink, and here red cabbage and soap solution, so you have colored here yellowish green. And this is the most important part of this practical. It is the observation table, what you have observed from your experiments. Like here it is the indicator turmeric, china rose, red cabbage. It will happen in acidic solution, what it will happen in basic and in neutral solution. You have to observe and write the observation table. Now this is the most important one. And one thing to remember, don't use pen or marker or sketch pens on this page. You have just to use pencils, and if you do color here you have to use pencil color or crayons. Don't use sketch pens and all, or watercolor. Don't use here, just use pencil colors. Now here you have to write, first of all you have to write the aim. What is your aim of your project? Here the aim is to observe the change in color of natural indicators turmeric, china rose and red cabbage in acidic, basic and neutral solution. So this is the aim, what you have to show, what you have to find. Now what are the materials you require, you have to write here. And mind this thing that you have to write this with black pen so that it will highlight the name. And this you have to write in blue pen, and this is the heading, so you have to write in capital letters. And here is the materials required, what you require. You can give here bullet points, indicators, turmeric powder, china rose petals. You can see from here what are the solutions, what are the apparatus, means what are the things you require to perform this experiment, like test tubes, droppers, beakers, warm water. Now procedure, what you have done, how you have done. So this is the procedure, and here you don't have to use points, you have to use your numbers because it is going stepwise. So the first procedure what you have done is natural indicators were prepared by extracting colors from china rose petals and red cabbage using warm water. You can see and read from this table. Now moving to the next page. You don't have to draw here anything, you just use this page and give here page number 2, and you can continue here. Now this is observation, what you have observed. Here you have to write that the changes in color for each natural indicator in acidic, basic and neutral solutions were observed and recorded systematically. Refer to the observation table provided on the first blank page of this experiment. So they will refer to the first blank page of this experiment, means this observation table. It is talking about this observation table. Now then you have to write here the interference, means what is the conclusion, what you got the information from this experiment. First is you have to give your numbers. First is, it was concluded that turmeric serves as a base indicator as it turns reddish brown in basic solution. Then China rose was found to turn dark pink in acid and green in bases. Red cabbage was observed to be a versatile indicator, turning red in acids and green yellow in bases. Now these three are the main interference, main results. So these three test tubes you have to draw here. We have drawn here just these two, so these three things. For example, here we got reddish brown in basic solution. It was of turmeric. So turmeric plus soap, reddish brown. Then you have to move to the precautions. What precaution you have to take? First precaution. Here you have to use bullet points. Clean test tubes were used for each test to prevent contamination. Then droppers were rinsed before switching between different indicators. Chemicals were handled carefully to avoid contact with skins. Means what are the precautions you have taken from here? What are the safety measures you have taken while performing these indicators? And remember to write this all the things in third person form, like uh, indicators were prepared. Means you don't have to write that prepare the indicators, you have to write indicators were prepared, means the things are done by you, so you have to mention in that way. Now this experiment is completed. Now the next experiment, which is of biology. So first of all you have to write here now this process is about the anatomy of hibiscus flower. So you have to draw here the longitudinal section of hibiscus flower. Then you have to draw this diagram. This diagram is there in your book, so you can copy paste from the book with the pencil. Now parts of hibiscus flower, sepals, you can give here heading sepals and write about the sepals, and similar process you can continue here. And in the stamen you will get two main points, which are first the anther. So you have to give your subheading with blue pen. Okay, you don't have to use your black pen because this is subheading, so blue pen. Then similarly you don't have to draw here anything because there is nothing to draw here. You have to draw here because you have mentioned the part of the flower, hibiscus flower here, so you have to draw here. Now you don't have to draw anything here. And okay, the same thing you have to draw here. Now reproductive system, you have to explain little bit. Then point wise you have to write, like the first point is pollination, you can see from this table. Now here you have to write here, and germination, fertilization. Here germination is written, so you can draw this drawing of germination of pollen on stigma from your book. It is given on your book, this diagram, so you can copy paste from here, from there, and you can write here. And seeds and fruit development. Okay, now finish, your practical is finished. Now you can leave these pages simply. So this was for today. Thank you for watching till the end.